All righty. So today we are going to be talking about Chapter 6, viruses and prions, okay? And specifically, we're going to cover the structure of viruses and, the mutation rates of the different types of viruses. Okay, so here's an overview of what we're going to be covering today. We're going to go over the learning objectives. I'll answer any questions you may have. Then we're going to review cell structure and then go from there into viral cell structures. Okay, so we're going to compare and contrast the two. And then we're going to talk about DNA polymerase versus RNA polymerase. So this is going to be a bit of a review from last week. Then we'll talk about prions, and then we'll talk about what to do for next time. All right, so this chapter helps us move toward being able to meet the following learning objectives. Be able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify key agents and processes of microbial information transfer, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. All right, any questions?
I do, but not about the chapter.
Sure.
I have a quick question about the final. So I was printing off all of the exam, or trying to get them all, the study guides all printed off. And didn't you say that the final exam was basically going to be the last few chapters, or is it a compilation of everything we've learned in the class?
So it's going to be the last 3 chapters.
Okay, all right, because just glancing at the study guide, it led me to believe different. So I wanted to double check. Okay, thank you.
I will have to go in and make sure I don't have the wrong study guide. Because it used to be that it would be it was a comprehensive final. Not anymore. So
thank you.
Yeah, quite welcome. So I will actually, let me make a note to check that. Okay. All right, other questions?
Are we going to get feedback on the case study 1 before case study 2 is done?
Actually, no. As you can tell, I've gotten very behind on grading. Unfortunately, my apologies. I have never been this far behind on grading ever. This is a new low for this semester. So my plan is to give you extensive feedback. But I'm planning on getting those graded this week. And to do that, to be able to get those graded by the end of this week, I'm not going to have time to give extensive feedback. So instead, what I'm going to do is you have the answers. The answers are in week 4. And then once I grade it, you'll see the rubric that I use to grade with, and it's going to be the same rubric that I use for all of the case studies. And so between comparing the grading rubric and the answers, that should give you an idea of where you can improve for case study number 2. Okay. And then hopefully I will be back up just to, caught up. And then I'll be able to give you extensive comments on case study number 2.
Okay, thank you.
Oh, quite welcome. Okay, other questions? Okay, if you do think of some, let me know. I'm happy to answer them. But in the meantime, we're going to talk about cell structure versus viral structure. So let us go to the whiteboard. All right. First off, okay, we're going to review cell structure. Now, this is going to be just standard cell structure. I mean, we can definitely talk about eukaryotic versus prokaryotic structure, but we're going to talk about mainly structures that all cells have. What do you have to be considered a cell? Okay. All right. So what are some structures that you have to be considered a cell?
Nucleus?
Yes. Let's go ahead and put nucleus down. You do need a nucleus if you are what kind of cell?
Eukaryotic, right?
Very good. Yes. So let's go ahead and get a eukaryotic versus prokaryotic. But do all cells have nucleus? Have a nucleus?
No, prokaryotes don't.
Yeah, very good. Whoops, I just drew a line right through my eukaryote there. Okay, so, but a nucleus is something that we use to identify eukaryotic cells versus prokaryotic cells. So, very good. Okay. All right, so what is another structure that cells have that both eukaryotes and, sorry, what?
Phospholipid bilayer.
Okay, phospholipid bilayer forming what?
The plasma membrane?
Yeah, the plasma membrane. Sorry.
No worries. Very good. Yes. So all cells have a cell or plasma membrane. And it is composed of phospholipids. Okay, so it is a phospholipid bilayer. Okay. Make it a little interesting to read when I go around the corner here. All right, so what else do all cells have?
Do they have either DNA or RNA, but not both, or is that viruses?
That is viruses. So go ahead and restate that for cells.
They have DNA?
They do have DNA, okay? So I'm going to go ahead and do DNA in blue and tell you what, we'll make this a prokaryotic cell. We'll make this a circular DNA. If I were doing a eukaryotic cell, it would be a linear DNA, okay? So the DNA makes up a fair amount of what structure in the cell?
The nuclear envelope.
If it is a eukaryote, you do have a nuclear envelope around it.
Or is it the
to protect it.
nucleolus?
In a eukaryote, we would have a nucleolus. So in a eukaryotic cell, we're going to put this in the background. So I would have a nucleus, and then when we stain a nucleus, we have a darker staining area that we call the nucleolus. Okay? And that's an area where we're doing a lot of transcription. Okay? But that would be in just eukaryotes. Okay? What do we have that's in the nucleus in eukaryotes that's in the nucleoid region?
Plasm? Oh, I didn't let you finish, but don't cells have cytoplasm?
Oh, yes, they all have cytoplasm. Let's go ahead and put cytoplasm here.
Are you looking for the word chromosomes?
I am, yes. Yeah, so DNA plus protein make up the chromosome, okay? And the chromosome in cells, okay, is always composed of DNA, okay, and protein. Okay, now we do, well, and then we can also call this the genome. And a number of different things that we can call this, okay? All right, so we also have RNA in a cell. Give me 3 types of RNA.
mRNA, rRNA, and tRNA.
Very good. Let's start with mRNA. Okay.
It's messenger, right?
Very good. Yes, we have messenger RNA. And what does messenger RNA do? Or what is it?
Isn't it the RNA that the DNA codes onto.
Yes, yes. So it is a copy of a gene, okay, from the chromosome. Okay. You know what? Genome is not showing up real well here, so let's do this in black. Okay. So the mRNA is a copy. Okay. All right. So what about ribosomal RNA? Where do we find that? I mean, this is a dust statement.
Ribosomes.
Ribosomes. Okay. Ribosomes are made up of rRNA or ribosomal RNA. Okay. So I would transcribe ribosomal RNA off of my genome and put it together with proteins to make a ribosome. Okay, what do ribosomes do? What's their job in the cell?
Synthesize protein.
Very good. They
Yep.
synthesize proteins. So they clamp onto this mRNA and they read the information in it and make proteins. Okay. All right. So the third one, tRNAs. Let's go ahead and put tRNAs over here. Okay.
They're the transfer.
They're the transfer. And what do they transfer?
Amino acids to the ribosome.
Very good. Yes, exactly. They transfer amino acids to the ribosome. Good job. All right. So in the cell, we have both DNA in the chromosome and we have RNA. Now in this class, we're only going to go over 3 different kinds of RNA. In A and P, you'll learn about others. But we have the mRNA, we have the ribosomal RNA, and the tRNA. Okay. All right. So what is something else that all cells have to have? It relates to the plasma membrane.
Protein.
Yes, and we have proteins in the plasma membrane. Specifically, what kind of proteins?
Transfer proteins.
Very good, transfer proteins. If we did not have transfer proteins, we'd have no way of getting stuff from the outside to the inside and no way of getting stuff from the inside to the outside. Okay, so we've got transfer, it's supposed to be an F, or transport. Okay, so this is, now there's lots of other things inside of a cell, okay, or that make up a cell, but these are things that we see in all types of cells, whether we're talking about a eukaryote or a prokaryote, okay. All right, so this is your basic cell structure. Excuse me. Now we're going to talk about viruses. Okay. All right. So it was already mentioned, but viruses have either what or what?
DNA or RNA.
Very good. They have either DNA or RNA for their genomes. Okay. So that's kind of weird because cells have both. They have both and they need both. If we were to take all the tRNAs out, the cell would not be able to make proteins and it would die. Okay? If we took the DNA genome out, it would not be able to make mRNAs, so it would not be able to make proteins, so it would die. Okay? But viruses are weird. Okay? They have genomes and they're composed of either DNA or RNA, okay? But not both. If I were to take a virus, lyse it, extract out the nucleic acids, I would find one or the other, okay? But if I do the same thing with the cell, I find both, okay?
And they can't
Alright.
necessarily replicate on their own, correct? They need the host to be able to do that.
That is correct. They do need a host because they don't have all of the stuff that's needed to make their own proteins. Okay, but they do have a genome. They have a genome that can take over some of the metabolic equipment, if you want to think of it that way. Okay, they can be recognized by ribosomes. They can be recognized by DNA polymerases and different things that are inside of the cell. Yeah. So exactly. Okay, now the world outside of a cell is kind of dangerous for DNA and RNA. Okay, there's all sorts of things that are going to break it down. So all viruses, okay, have a capsid. Okay, so a
But
capsid.
the protein coat that's around it.
Very good. Yes, it is a protein coat.
Okay, I had that written down and not the capsid in my notes, but I'll fix that so I know it's both.
Well, the protein coat is what we call we call it a capsid.
Yeah.
Yeah. Okay, now a third thing that all viruses have to have is some method of grabbing a hold of the cell. Okay.
Spikes.
Yes, on most viruses it is spikes. Now, there are a few viruses that have little divots in their capsids that host cells receptors go into. Okay, but those are the exceptions. You don't need to worry about it for this class. For this class, remember that there are spikes, which are proteins that attach to the host cell. Okay. Okay. All viruses. Okay. All viruses have these three things. Okay. Now you can see that a virus is very pared down. Okay. From a cell. And I didn't leave myself very much room, but let's go ahead and start drawing one. So I'm going to do over here, I'm going to do a non-enveloped virus. Okay. So I'm going to go ahead and draw the capsid in brown. Okay. And the capsids come in, for this class, 3 different kinds. Okay. This hexagon that I've drawn here is a cartoon for what's called an icosahedral capsid. Okay, so it's kind of round, but it's more like a 20-sided die. So for those of you that play role-playing games, you know exactly what I'm talking about. And for those of you that don't play role-playing games, you're going, oh no, we have descended into nerdiness, which is true. Okay, so this is the capsid. And because it's non-enveloped, my spikes are going to be coming off this capsid. Okay, they are going to be a part of the capsid. Okay, so these are our spikes. And the spikes are what grab a hold of the host cell. Okay. All right, and then this one, I'm going to make a DNA virus. Okay, so its genome is made up of DNA. Okay. Okay, so that is your very simplest non-enveloped virus. Okay. All right, so I'm going to erase this so that we have room for me to draw an enveloped virus. Okay. Okay, so just for the fun of it, I'm going to do a different capsid shape. Okay. Now, the capsid shape is not unique to either non-enveloped or enveloped. Okay. It can be in either kind. Okay, but this is a roundish icosahedral one. For the enveloped virus, I'm going to do what we call a helical one. And what it is the proteins that make up the capsid kind of form, whoops, that's scary. There we go, that's better. Okay, kind of form a helix. So as they're coming together, they do this kind of thing. And they form a tube. Okay. So here's an open end, here's the closed end. Okay. So in this situation, okay, this is a helical capsid. And oftentimes they will bend like this. Okay. Or since I'm doing Ebola, let me try this again. Ebola is really long and it's helical, but it kind of wraps around itself and kind of, they call it forming a shepherd's prick. We'll draw like this. Okay, so it kind of forms this flexible tube. Okay, and we call that a helical capsid. Now, Ebola is an RNA virus, so I'm going to go ahead and put an RNA genome in there. Okay. So it has a genome, but it's made up of RNA. And just because I'm lazy, I'm not going to draw it all the way. Okay. And it has spikes. Okay. But these spikes are not a part of the protein capsid. Okay. I'm showing them floating out here, but what is happening is that they are embedded in a phospholipid bilayer, okay, that I'm going to draw here. Yep. That coats the outside of the capsid. Okay, and so these spikes are embedded in the envelope. Okay. Now this envelope came from the last host cell. Okay. So with this host cell, if it had a virus that was an enveloped virus, and it's more complicated than this, but I'm going to overgeneralize to keep it simple. Okay. As the virus is replicating, and we're going to talk more about this on next time, but they kind of go bleep. Okay, as the virus is coming out, okay, so it's coming out with its capsid, and they kind of pinch off, okay, and when they do, they pinch off with a part of the host cell membrane that we call an envelope, okay, and previous to this, the spikes were embedded in the host cell plasma membrane, and that's how the virus gets its spikes. Okay.
So is a non-enveloped virus a virus that hasn't gotten to a host cell yet?
That is a very good question. No, it is a virus that does not need an envelope. Okay, that's not part of its genetic makeup. So let's say that I have my non-enveloped virus here. Okay, it's all ready to go. It's got its DNA inside of it. This poor person has two different viruses going on. One of them being Ebola. They're really sick. Okay, so what happens is two different things can happen. Either the virus can stimulate the cell to pop, and then it gets released into the outside, okay, as a non-enveloped, or sometimes we call them naked viruses. Okay. And so they don't have an envelope, or they will trick the cell into exporting it. Okay, so instead of budding out like this, it'll be exocytosed. Okay, questions? Yes.
The envelope is the plasma membrane around it.
Yes.
Okay. All right.
Yeah.
Cool.
But once it's no longer a part of the cell, we don't call it a plasma membrane anymore. And there are no transporter proteins or any of the other proteins that would be recognizable
So we call it
a part of this cell.
So we just call it an envelope.
Yeah, so we call it an envelope to distinguish it from a plasma or cell membrane. All right, so an envelope is something that some viruses have, but not all of them have. Okay. Another thing that some viruses have, but not all of them have, okay, are viral specific enzymes that get packaged inside of the capsid. Okay. So let's suppose that this virus needs an enzyme that the cell doesn't have. It's got to pack it with it. Okay. So some viruses have viral specific enzymes that are packaged inside of the capsid, okay, and some don't. Okay. But once they get into the cell, and let's go ahead and go with this DNA virus. Okay. Once they get in, then they're going to trick the cell into thinking that this is part of the cell DNA. And so mRNAs are going to start being transcribed. And then these viral mRNAs get taken to the ribosomes, and the ribosomes start making viral proteins, okay, that eventually will snap together, okay, to make the capsid and all of that, okay? So once they get into the cell, they do make viral specific enzymes, just not all of them get packaged into the capsid, okay? All right, questions? That's pretty much it, or as much as we're going to go into the structure of viruses for this class.
The little curly Q one there, what's it called?
Oh, this one, this is my cartoon of Ebola.
But there was another name you said for it.
Oh, it is a helical RNA virus.
Helical.
Yeah. Yeah.
Will we have to know the shape, like that name? or
Yes,
anything?
I do want you to know the three, for this class, 3 different kinds of capsid shapes. So there is the icosahedral or 20-sided die shape. There's the helical shape, okay? And they don't always bend around like this. If I were to have gone with rabies instead, the helical capsid bends in a U shape, and then the envelope comes around like this, and they are bullet shaped, which I find horribly ironic. Because that's what we used to have to do to rabid dogs, is shoot them.
I apologize. What was the 20-sided dice one called?
Oh, icosahedral. Let's see if I can spell it right.
Icosahedral.
That's how you pronounce it. I was going through my notes and I had.
Yeah,
no-
that's not how I was pronouncing it.
Yeah, that's how I've heard it pronounced. So I hope it's correct. Yep. So yeah, so it's basically like a 20-sided die. Yeah. Okay, now there is a third shape of capsid. Okay. It's the only other one you need to know for this class, but we call it complex. So let's say I've got a bacterial virus, okay, a bacterial phage. It's got an icosahedral head. It's got a helical shaft, and then its spikes are these funky little leg things. And so it ends up looking like a little lunar landing module. Okay. And we package the DNA inside. Okay. And so we call that a complex capsid. And you can kind of see why we call it complex, because we've taken 2 other shapes and combined them to make something that is, unique. Okay,
Wait.
question. Oh, yeah.
To go over that again, it's like this weird bug-shaped thing in the top right corner.
Yeah. Yeah, we call this a complex. Oh, and I have gone to the edge of my whiteboard and I can't write up there, at least not all the way. Yeah, we would call this a complex capsid. Okay. Because, you know, it kind of combines the shapes of the more simple capsids, okay, to make this complex shape. Yeah, next time we meet, we're going to be talking about this particular little bug looking guy. Okay, other questions?
Your
And
drawing,
what,
your drawings don't look the same like they do in the book. So that's why it's kind of like, what the heck?
But if you were to draw out the drawings in the book, they would look similar. Actually, I think for the helical, I think they did them straight, but they sometimes they do bend around.
what was the name of the second one again? I totally forgot.
Oh, this one helical.
Do we need to know your shapes or the book shapes or both?
My shapes are the book shapes.
Okay, so we'll
Yeah,
see.
if you take a close look at this versus the book, there you're going to find that mine is the books ones are better, but you know, my cartoons are pretty close to them. Yeah, believe it or not. Okay, other questions? Okay, if I completely buried you, hopefully not. Okay, while we're here, actually, I'm gonna clear the drawings, or if you're ready for me to do that, and I am recording this, so I can go back and look. Okay, so when we are classifying viruses, okay. So to be a virus, you've got to have those three things. And just for the fun of it, what are those three things? What are the three things that you have to be a virus?
You have to have either DNA or RNA, but not both.
Very good. Okay.
Capsid.
Okay, a capsid
Spikes.
and spikes. Okay. And the reason why I bring that up again is because we're going to get into things that are close to viruses, but not quite. Okay. So the first thing that we do is when we are classifying viruses, is we divide them based on host. So I can have bacterial viruses. Okay. And I can have viruses that infect animals. And that's what we're going to talk about in this class. But you can also have viruses that infect plants. And I can have viruses that infect fungi and protozoa. And what we have recently found is that there are even viruses that infect other viruses, which is kind of funky. So in other words, viruses have evolved to infect every kind of cellular life and even parasitize their own kind of bioactive molecular. I'm not
How
sure
does
what they call them.
that work? Like, what viruses attack other viruses?
I know, isn't that creepy? Okay, so I'm going to go ahead and now, like I said, this is fairly recent. We didn't think that viruses could do this to each other. But let's say I look under an electron microscope and I've got a virus here. And just for the fun of it, let's make it a DNA virus. Okay. And with some viruses, we have seen some with another little capsid inside, like they're pregnant. And this is the virus that has infected another virus, okay? So it's kind of like it's hitching a ride. And then when it gets to the host cell, both of them get replicated. But this one takes energy away from this one if it were not infected with a parasitic virus. So it slows down the ability of this host virus to make as much of itself as if it were not infected.
So that kind of sounds like a good thing, maybe?
Oh, it's a good thing for us. Yeah. Well, unless you get infected with both of them. Usually it causes more severe disease.
So the virus that's inside the other virus, it won't be able to replicate until it finds a host.
Until the host virus finds a host.
Okay, okay. Thank you.
What kind of
know.
viruses do that? Like what kind of example viruses do that?
You know what? They are so new that we're still figuring this out. Okay. So far as I know, there are no animal viruses that have parasitic viruses or viruses that parasitize them that we know of. Now that we know to look for them, we're probably going to find more.
That is interesting.
Yeah, we've found them in protozoa, and I think we've found them in fungi. Yeah, isn't that funky? Yeah.
Are we sure we didn't just find out that viruses can actually just be pregnant and it's not really infected by another virus?
You would think it sure looks like a pregnant virus, doesn't it? But no, when we separate them out, this one has a different genome than this one. And in fact, sometimes they have a different kind of genome. So some of them, you know, like it'll be an RNA virus inside of a DNA virus. Yeah, isn't that funky? Yeah, so viruses, they don't care. They'll parasitize anything. But they are very specific for their host cells. So animal viruses, and I'm going to generalize, animal viruses do not infect protozoa, and protozoal viruses do not infect fungi or any of the others. Okay, so they're very, very specific. They want just one specific host, which is why this is the first thing that we separate them into, is based on host. So we've got bacterial viruses that we call phages. Okay. And then animal viruses and protozoan viruses and that kind of thing. Okay. So just for the sake of simplicity, I'm going to start over here just so I don't get too cluttered. The next thing that we separate them into is DNA versus RNA. Okay. And then from there, we will talk about shape, if they're enveloped or not. Okay. What their genome is. Okay, so is it, we can have single-stranded DNA viruses, we can have double-stranded DNA viruses. Okay, so those are the first two big divisions, though, is the host, and then whether they're DNA and or RNA. And then we'll go into the shape of the capsid. We'll go into whether they're enveloped or not. Okay, so just for the fun of it, let's talk about the virus that has been plaguing us these last 2 years. Okay, and I'll go ahead and label it. The virus that causes COVID is called SARS-CoV-2. Okay, so CoV is coronavirus. That's short for coronavirus, and SARS is severe. I forget what the A stands for. Severe adult respiratory syndrome. Sure, we'll say that. I'm not sure if that's what the A stands for. Okay, so it is an icosahedral capsid, okay, and it is enveloped. And this becomes important later on when we talk about control of microbes and other pathogens like viruses. in the environment. And then the spikes are really big. And they've got a visible knob on the end when we look at them under an electron microscope, which is why we call them coronaviruses, because it looks like it's got a crown. Okay, the crown is the spike. And then we have an RNA genome inside. So SARS-CoV-2 is an animal virus because it infects humans, and it's an RNA enveloped virus. Okay. Hey, the questions. And by the way, I could draw the same thing over here for all of these.
Isn't SARS also kind of like different types of influenza?
No, influenza is very, very similar. It's also an animal RNA enveloped virus, but they're not closely related. When we sequence their RNA genomes, they're very different. But they're both respiratory viruses, so we often talk about them because preventing transmission is very similar because they go from person to person in a similar way. But influenza has these mushroom-shaped spikes that we call hemagglutinin, so they're H or HA. And then we've got these funky, more column-like ones.
Those
we
that
call-
NA?
Those are the NA. So when we, this is a few years back now, I haven't been putting my mushrooms on. When we talk about H1N1, we're talking about these types of spikes. And then whereas SARS-CoV-2 has one strand for its genome, influenza has 8 segments, okay, of RNA. And so we end up with what's called recombination. So if I get 2 types of influenza infecting the same cell, these segments can sort, and so I'll get some of one in one and some of one in the other, and then I get big changes in these spike proteins, and it usually ends up in a more severe disease. Because like with the case of H1N1, we see that in bird influenza, but we didn't see it in human influenza before, I want to say 2012, 2000, something around in that range. And so people were getting really sick, okay, because it was not a slight change, okay, in the normal human influenza that we were seeing, okay? And that's kind of what happened with SARS-CoV-2. Most coronaviruses cause the common cold. And it's because they've been in humans for a long time. And so we have plenty of exposure to it. And also it's also gotten less virulent. It causes less severe disease. And so for most coronaviruses, okay, they do not cause severe disease. But because the SARS family of coronaviruses have jumped recently from non-human animals, that's why we're seeing such severe disease, okay?
Because of the mutation and us not being able to handle it, basically.
Yeah, it's spikes that we have not seen before as a human population, and so we have a delay in our immune system responding to it, and so that usually ends up in more severe disease. Now we had some people who are asymptomatic; they got lucky. Yeah. Okay, other questions? Okay, if you think of other questions, let me know. In the meantime, I'm going to go back to the PowerPoint so I can make sure I did not forget something. Okay, so we did talk about cell structure versus viral structure. Okay. And the next thing is talking about DNA polymerase versus RNA polymerase and why we care, why I'm bringing that up in a discussion about viruses. Okay. All right. So remember, we separate viruses into either those that have a DNA genome or those that have an RNA genome. Okay. So what is the enzyme that will make a DNA copy from a DNA template? So the enzyme that allows us to replicate DNA.
Is that one helicase?
That one is a part of it. Yes.
Polymerase? Then?
Yes, polymerase. And because we're making DNA, we call this a DNA polymerase. We shorten it to just DNA polymerase. Okay. Okay, so when I'm making an RNA copy of DNA, okay, so we're talking transcription here. In cells, what enzyme transcribes DNA into, say, an mRNA or a tRNA or an rRNA?
Repeat the question.
Okay, so if DNA polymerase makes DNA, what enzyme would make RNA from a DNA template? What enzyme transcribes DNA?
RNA polymerase?
Very good, RNA polymerase. Okay. All right. So the RNA, okay, let's talk about their jobs. Let me get rid of some of this here so I've got rid. Okay, so DNA polymerases, okay, replicate the DNA. Okay, so I'm making a complete copy of the genome. Okay, what happens when a mistake happens and it doesn't get fixed? And then it proceeds to go on to progeny or child cells.
Mutation?
Very good. Yes. So if the DNA polymerase makes a mistake that does not get caught, it leads to a mutation. Okay. Now, generally speaking, mutations are bad. They do not benefit the cell. Okay. Very rarely you will get a mutation that does provide a benefit to the cell, and it will proceed to have more babies than other cells. Okay. But usually mutations cause harm to the cell. Okay. So DNA polymerase has a high level of proofreading. Okay. And so let's say that I have a strand of DNA that I'm replicating. So I'm going to get my replication port going here. Okay. And we'll do the new strand in purple just for the fun of it. Okay, and we will say that I've got A, T, G, C, and just because I'm lazy, okay. All right, so with the new strand, okay, with the DNA polymerase, what goes with A?
In DNA?
Yeah,
T.
very good. And what goes with T?
A.
Okay. And let's go ahead and draw my DNA polymerase here. Okay. Now let's say that accidentally, okay, another A got in here, and the DNA polymerase was in kind of a hurry, and it attached this A to the other A when it should have been a C. Okay. The DNA polymerase will look back and make sure that it's made the right choice, okay, in the synthesis. And it's going to look back and say, oh, that's not right. I'm going to go back and go snip, 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 and I'm going to try again, okay? And it keeps trying until it gets a C. And then it moves on, and it'll put a G here and so on. We call that the proofreading function of DNA polymerase, okay? So the mutation rate is really very, 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 very low, okay? We do not get mutations very often. Now with bacteria, we tend to see more mutations within the population just because they divide so frequently. Okay, so RNA polymerase, on the other hand, let's go ahead and let's get a DNA bubble here where instead of making DNA, I'm making an RNA transcript. Okay. And we will get the same nucleotides going. Okay, so this would be T, A, C, T. Oh, let's see. That would be T, A. Okay. So this one I was showing you synthesis of this particular strand. Now, just because I'm used to drawing the mRNAs flopping off. Okay. Now, let's say that RNA polymerase, okay, instead of putting a C here, accidentally puts an A. Okay. It does the same thing, okay, that DNA polymerase does, but it doesn't do it as well. So let's say that, let's go ahead and switch colors here so it's easier to see. So I had, you know, a G here like I should. I had a U here and an A and on down the line, okay? And I accidentally get an A here, but then I get an A here like I should, and I get a U here and so on. If it doesn't catch that mistake, it's not a big deal. Okay? Maybe it'll make an incorrect protein, but this is mRNAs are temporary copies. So after it gets transcribed a certain number of times, nucleases, enzymes that go snip, 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 snip on these mRNAs will break that up and the cell can try again. So maybe it gets a few misfolded proteins. It's not that big of a deal. RNA polymerase will probably get it right on the next go round. Okay. So RNA polymerase has a lower level of proofreading. Okay, than DNA polymerase. Okay. So why do we care? It leads to the mutation rate difference that we see in DNA viruses versus RNA viruses, okay? Since we started being able, so over here on the DNA side, okay, I drew the influenza virus over here, but it really is an RNA virus. Okay, so the DNA virus family that I'm most familiar with is the herpes family, okay? And since we started isolating and characterizing viruses, okay, long about the 40s, we have not seen a lot of change between herpes simplex 1, herpes simplex 2, and varicella zoster virus. That's the one that causes chickenpox, okay? So these are very closely related viruses. When we take their DNA and we look at the order of it, we can say, oh yeah, these are like siblings, okay? Came from the same parent, okay? In the nearly, I want to say about 80 years okay, that we've been sequencing. Well, actually, it's actually been, yeah, it's been since the 90s since we've been actually sequencing them. But as far as telling the difference between these, they're very closely related. There hasn't been a lot of difference showing up among these three viruses, okay, in about the approximately 100 years since we've had the start of virology and the study of viruses. SARS, on the other hand, SARS-CoV-2, we have seen in 2 years, we have seen 3 different serotypes pop up that are different enough that they're like the difference between HSV1 and HSV2. Okay. And that's in 2 years. Okay. So we've had, well, the first one, we went back and named it after the fact. It's the Alpha variant. And then we got Delta. And then we got Omicron. Okay. And it's because this is an RNA virus. The DNA polymerase doesn't do as good a job, so we get a whole lot more mutations, we get a lot more variation. These 3 are the ones that got mutations that allowed them to outcompete the other serotypes. Okay, they just did better, and that's why they spread. Whereas, say I get a mutation where I don't have as good a spikes, they don't grab a hold of human respiratory cells as well, we're not going to get more of those. They're going to get eliminated. Okay. Kind of the same thing with influenza. Every year we have little changes in these spikes. And that is why we have to, we put out a new influenza vaccine every year. Okay. And it's because of the higher mutation rate, because we're talking about an RNA genome. And that means that we're talking about RNA polymerase that makes the genome. Okay. So if SARS-CoV-2 had been a DNA virus, we would not have these variants. Yay, right? Okay, questions about DNA polymerase versus RNA polymerase and why we care when it comes to talking about viruses. Okay, let's go on to our next thing. All right, so we have viruses. And all viruses have to have a capsid, spikes, and either an RNA or a DNA genome. Now we have pathogens, okay, subcellular or acellular, intracellular parasites, okay, that are missing some of these pieces. Okay, we're going to go over them very briefly. So the first one is a virioid. Okay, oid in, I think I got that backwards. Oid, that looks better, means like. Okay, so like in science fiction shows, when they talk about humanoid aliens, that means they're human-like. They're not human, but they sure look like them. All right, so virioids, okay, do have a genome. They have an RNA genome. But that's it. Okay, they are these little circles of RNA that we find infecting plants. Oops. And some of them are important in agriculture, which is why we care. Okay? So virioids do not have a capsid. No capsid. Okay. And there are no spikes. If they had spikes, they'd have no place to put them. Okay. So They're just these little circles of RNA that go from cell to cell. Now you're probably saying, and which when I first heard about varioids, I went, how do they get from cell to cell? Once they get out into the environment, they're going to get broken down. Okay. Well, what happens is they get passed from plant to plant 2 different ways. Say an infected branch on a tree rubs against its neighbor's branch and causes a wound, then these varioids get transferred directly from infected cell to infected cell. Okay, so it's through abrasion. Okay. Another one is if you have a bug that bites an infected plant and then goes and bites a non-infected plant, it can carry that varioid kind of like a virus, like a mosquito does for us. Okay. Now there's another one that was in chapter 5, but we didn't talk about, and that's a plasmid. And a plasmid is a circle of DNA. Okay. They tend to be bigger than the varioids, okay, so that they have a DNA genome. Okay, but once again, no capsid, no spikes. Okay, so how do they get from cell to cell? It's one of the 5 transes. You're going, I don't remember this at all. Okay, they can be released briefly by a dead cell, okay, and then a new cell takes them up out of the environment, okay. We do that in the lab a lot, okay. And then the other one is conjugation. Okay, so a pilus or more than one pili, okay, a protein tube between 2 different cells, okay. So I'm going to get a, let's say for the fun of it, I'm going to do a gram-positive cell. Okay, if I have a plasmid, it has a gene for forming this pilus, okay, that's a hollow tube that's going to grab a hold of another cell, okay, and then this plasmid is going to replicate itself and come over to this other cell. Okay. All right, how are we doing so far?
And so both of these act as a virus. Is that
They act like a virus, but they're not viruses because they don't have a capsid, they don't have spikes.
okay?
But they are pathogens, okay? Well, except for the plasmid, actually, they do slow down the replication. So they kind of make, so the bacterium doesn't feel so good, but sometimes they will carry a gene for antibiotic resistance. So they actually confer a benefit. Yeah. We wish that they wouldn't do that with bacteria because it makes our lives harder, but it actually, in the end, benefits the bacteria. All right. So now we're going to go on to prions. Okay. So prions do not have a nucleic acid genome. Okay, they don't have a capsid, they don't have a spike. They are prions, are infectious proteins. Okay. And it took us decades, okay, to believe the data that indicated that they were just proteins, okay, because proteins, how can proteins be infectious? So let us compare, and I'm going to start out with venom, okay, which is a poisonous protein most of the time. Okay. And then we're going to compare that to a prion. Okay. So let's say I'm out hiking. Okay. And, you know, I get too close to Middlester rattlesnake here and I get bit. Okay. So what happens on a cellular level? Okay. Oh, let's do this in red because rattlesnake venom pops red blood cells. Okay. So these red blood cells, the venom gets injected, okay, and it forms pores in the red blood cell and it causes it to lyse. Okay. So we call rattlesnake venom hemolytic venom. Okay. If we were talking cobras, then it would specifically attack nerve cells. Okay. But since I was lucky and I got bit on my ankle, it's pretty much going to stay there unless it gets into the blood and then it might circulate. But, you know, it's got a long way to go to get to my heart to cause problems. Okay. If I got bit on my arm, I'm more worried about it. Okay. But these proteins don't make more of themselves. Okay. So they dilute. Okay, themselves. Okay, we see dilution of the venom. It's more severe at the bite site, less severe around, less severe around. Okay, so with poisons, with venoms, we do see dilution. Okay, with prions, we do not. Okay, so let's go ahead and get my very scary drawing of a neuron. Yeah. That kind of looks like a neuron, right? Yep. Okay, so what happens with prions is that prions mimic normal cellular proteins, okay, that are expressed on the outside of the cell. Okay, they're made in the cell, but they get sent to the outside of the neuron. Okay, prions are a misfolded version of this. Okay. And they're kind of U-shaped and they wrap around and they change the shape, okay, of the normal cellular protein into this other kind of banana shape, okay? And so they stop doing their job. So the cell says, hey, you're not doing your job. We're going to bring you in and break you down, okay? So we call this an endosome, and we get a bunch of these U-shaped prions in there, and we do not have the enzymes to break them down. So the cell keeps trying to bring in these misfunctioning, and I'm going to abbreviate my prions into C shapes for the sake of time. They keep bringing in these non-functioning, okay, prions, and they can't break them down, and eventually the cell chokes on them. Okay, but I'm getting more and more all the time. Okay, and so with prion diseases that are caused by prions, okay, the one you're probably most familiar with is mad cow disease. Okay, also known as, or more formally known as, spongiform encephalitis. Okay. Specifically, if it comes from cows, it's bovine. Spongiform encephalitis. Okay. What happens is with when we get patients who start showing problems with being able to walk, okay, they start having trouble speaking, and then eventually they lose cognitive function, and then they die. Okay. When we take a look at their brains, we see big holes. That's the spongiform. Okay, their brains look like sponges. Okay, it's because these prions have been accumulating in neurons, and the neurons have died. Okay, so with mad cow disease, okay, how we get it, okay, let's say I have eaten the meat from an infected cow that has these prions, okay. The prions are acid stable, and so they survive transit through my stomach. For whatever reason, our intestines say, hey, we need some of this. And they get sent up to your brain where they proceed to cause the problem, okay. There are inherited forms. That's how these prions get started, is I get a mutation in a gene for the cellular prion as opposed to the prion, okay? And so let's say that I have, in humans, we have 2 copies of each gene, okay? So I've got a copy from dad, and I've got a copy from mom, okay? And let's say dad has a mutation in this cellular protein, and it is a prion instead, okay? But mom's genes are making good proteins, okay? So over time, and it usually takes about 40 years for these prions to accumulate, and you end up with a spongiform type encephalitis, and then people die, okay? The most common one that's inherited is Crutchfield-Jacob disease, okay? The problem is with mad cow disease is you're getting a whole lot of prions all at once. And so it accelerates it. And so when people get mad cow disease or Crutchfield-Jacob disease variant, okay, generally we see that show up in about 2 to 10 years after the suspected ingestion. Okay, so it's accelerated. Okay. All right, questions about prions? Oh, by the way, we do have a prion in deer and elk in this area called chronic wasting disease. And so they're wanting, and we abbreviate that CWD. And so when you go hunting, they want, the health department wants to check your deer or your elk to make sure it does not have CWD. If you happen to shoot a very skinny, emaciated looking cow, deer species, okay, they start having problems being able to walk and chew, and so they lose weight. Yeah, make sure you get it checked and be careful while dressing it out, because you really don't want to pick up a prion. All right, any questions now that I've scared the living daylights out of you?
This class makes the world seem like a safe place.
It does, doesn't it?
So do you just have to ingest like the brain matter, or are the prions just everywhere?
From what we know is that it's mainly in the brain, but at least in Crutchfield-Jacob variant, it did get and circulate through the bloodstream temporarily, if nothing else. And so we're not quite sure how chronic wasting disease is transmitted. We think it's transmitted through nasal secretions, which makes sense. Your nose is kind of close to your brain. So yeah, you want to be very, very careful about nasal secretions in any deer or elk that's suspected of having CWD, because we're not exactly sure how transmission works with this one.
Okay, and prions, they like, what do they call, like they can capsulate, right? Or, you know, like, can they survive outside of, okay.
They can survive heat, so cooking doesn't help.
That's
They
what I
survive
had.
acid. Yeah,
Okay.
they can survive an acid bath of your stomach. Your cells cannot break them down. Our digestive enzymes do not break them down. They survive drying. Yeah, they're kind of scary.
What do they not survive?
Incineration. So if you take, in fact, that's what we do with cows when we find BSE in them, in the herd. We'll slaughter the entire herd and just burn, basically cremate them. So if you burn the prions to the point where the amino acids are separating from each other, then that'll take care of it. But who wants to eat a charcoal briquette? That's just to control the spread of the prions. Yeah. Okay, other questions? Okay, let us go to what to do for next time. Okay, I want to give you a preview of what's due on Friday. Okay, oh, by the way, here are the pictures from the book, which are much better than mine. But you notice I did do the, the little squares and the little U shapes. Okay, so yes, by the end of this week, I will have case study 1 done, but you may want to start working on case study 2. You've got 2 weeks to work on it. We do have exam 2 opening up this Friday. Okay. So, now's a good time to go visit with Rochelle, either on the discussion board or on her Zoom meetings, just to get some help with learning all of this stuff. Okay. And then if you haven't done so, go ahead and finish going through the contents of the week 7 lecture folder. Okay. All right, any questions?